Guys, we have Dr. Ruggieri here from Temple. He was initially in the urology department for 22 years, from 1990 to 2012, and now he's working in the anatomy department. He's here today to talk to us about CNS control of bowel and bladder function. He's got some cool research to talk about too in his talk as well, so try to listen closely and get as much as you can on the lecture. Thank you.
Well, thank you very much for having me up here. This is great. I've never spoken to such a full room in the last several years. It's great, except the medical school classroom. So it's great to have everybody here. I'm going to be speaking from the perspective not of an anatomist, but I spent about 22 years of my post-PhD training at Penn in the Pharmacology department at Temple as you know, NIH-funded investigator doing translational research. And I recently transferred to the Anatomy and Cell Biology department, where, as a basic science department, they were able to put me on what's called the tenure track. And as a 61-year-old man, I'm not sure how many rails of that track I have left, but I need to submit documentation of my teaching, and I'm going to send out an email for an evaluation. Very appreciate it if you fill it out, because last year's medical school class, only 16 out of 200 students filled them out. And one guy was really perturbed about everything. So I need a larger database if I can. But if you don't like what I talk, maybe you don't want to fill it out. That'd be good too. So I'm going to talk from the perspective of a pharmacologist and not an anatomist. So what's interesting to me about the gut is the enteric nervous system. And it's a pretty amazing system. This is from a book called Neurocontrol of Gastrointestinal Function that I looked at to prepare this talk. It digests between 100 and 700 tons of food throughout a lifetime. And this is a British book for an average British citizen. And that translates to 550 poultries. That's 550 chickens there, what you eat in a lifetime. 36 pigs, whole pigs. That's 36 pigs. That's what 36 pigs. British eat a lot of sheep, so 36 sheep. Eat a lot of mutton. And 8 oxen, full oxen. Some people like it better than the others. This guy seems to really like it. And 10,000 eggs. That's what 10,000 eggs looks like. And dairy products equivalent to 18 tons of milk. That's what 18 tons of milk look like. So the surface area is 100 meters square. So if we're at Lincoln Financial Field, that's what 100 square meters looks like from an individual's surface area of their GI tract that absorbs nutrients. It's in contact with 10 to the 14th bacteria, which are greater than the total number of human cells in the body by 10 to 1. And those bacteria do important functions. To understand how the enteric nervous system works, this is theoretically how you would approach it. You just describe the hardware, understand where the different neurons are and what they do. Describe how the wiring diagram is, how the synaptic interconnections are put together. And then the activity of each type of nerve doing various behaviors would need to be recorded. This actually is technically the most difficult thing to do. We know more about the central nervous system in the brain because it doesn't move when it performs its function than the gut, because every time you try to measure it, it's moving and you lose your measurement device. And then fourth, you have to model this on a computer to make it possible to prove that the understanding from the first 3 aims is sufficient to explain gut behavior. Now, we have some information on the first aim and a few preparations, some of them human, not all of them human. And some from step 2, the wiring diagram and the computer diagrams. But the third one has really not been achieved. And so essentially, we don't know much about how the enteric nervous system works. And that's really the challenge that I'm putting forth to you young people in the future, to try to understand how the enteric nervous system works. And that's where the discoveries are going to be made, I think, in the next decade or two. I've been working along with a gastroenterologist on gastroesophageal reflux, and we think we know a little bit more about nerves and how the enteric nervous system is involved. So I'm going to refer to that information a little bit. I'm not going to show all our data on GERD with Dr. Larry Miller, who's now up at Long Island Jewish Hospital in New York, who was at Temple for a long time before he moved. The innervation of the GI tract is, this shows the interconnections between the enteric nervous system and the central nervous system. It contains full reflex circuits intrinsic to the gut, the enteric motor neurons, and these are shown in blue in this diagram. And I'm going to, this is available to you. You can look this, it's going to be available on YouTube also. So the pathways project outwards via these intestinal fugal neurons in red. Is there a pointer I can use here? Oh yeah, okay. These intestinal fugal neurons shown in red here. And then to these yellow neurons, the CNS neurons in yellow, there's sympathetic ganglion, particularly in the gallbladder and pancreas. The sympathetic paravertebral ganglion in green receive both inputs from the CNS as well as the enteric nervous system. And this sensory information goes both to the back to the enteric nervous system between these intrinsic primary afferent and that are shown in purple, and to the CNS between the extrinsic primary afferents that are also shown in purple here. So so this is the basic wiring diagram. The major central nervous system innervation of the gut is from the 10th cranial nerve, the vagus nerve. But this is the diagram of what we know so far of AIM-1, the actual function, nerves that are there, and somewhat about how they're wired. It has ganglionated plexuses called the myenteric plexus between the longitudinal circular fibers, muscle fibers, shown in this diagram. And then nerve fiber bundles connect the ganglion and also form plexus that innervate the longitudinal muscle, circular muscle, the muscularis mucosa, which is lining right below the lamina propria, and intrinsic arteries and mucosa of the entire gut wall. Some of this is known for just some preparations, and some of them are mostly animal preparations, and it's not clear, and I think it's probably not true to assume that that what's known about these animal preparations definitely applies to human gut, and throughout the GI tract, it's probably not going to be the same. And this circuit diagram shows the control of motility in the small intestine. This is primarily data from the guinea pig small intestine. There are some data from humans showing that this is generally the way it works. It's actually very simplified, although it looks pretty complicated. It's really simplified from what it actually is. There's interconnected intrinsic sensory neurons, or known as intrinsic prime. That's these red cells here. And then there's these synapses with the descending, these ones in yellow, interneurons, as well as ascending interneurons that are shown in green, as well as the excitatory muscle motor neurons that are shown in blue here, and inhibitory neurons shown in purple. So it's a very complicated system, and how it actually works is really not that well developed. What we've shown for the gut, for the gastric lower, what's called the lower esophageal sphincter, but we think it really consists of at least 3 different components, which I'm not really able to get into too deeply. But the reason that relaxes, we think, is because there's an overactivation of nicotinic receptors in people with reflux. And that causes a release of inhibitory substance such as nitric oxide that then relaxes the smooth muscle of the lower esophageal high pressure zone. I'm deliberately not using the term lower esophageal sphincter because I think it's a misnomer, but that's a different lecture. So I'm going to just quickly go through classifications of a number of enteric neuropathies that you may run into and may probably treat here at Moss Rehab, such as Hirschsprung's disease, which is also known as colorectal aganglionosis, hypertrophic pyloric stenosis, multiple endocrine neoplasia, multiple intestinal dysplasia, and mitochondriopathies affecting the enteric nervous system. And here's a number of sporadic and acquired neuropathies. Everybody always talks about Chagas, but here in Philadelphia, it's unlikely that you'll ever see a case of Chagas, I think, but you have to learn about it. It's probably going to be on your boards. Neurogenic forms of intestinal pseudo-obstruction. There's some other much more poorly understood conditions, such as slow transit constipation, chronic constipation, including that associated with aging, pathogen-induced diarrhea. A little bit more is understood about this. And irritable bowel syndrome is very similar to a condition we've been looking at in urology for years at Temple, called interstitial cystitis. And I don't think we know a lot about either of those, but they're very similar. They have a lot of commonalities. And then autoimmune enteric neuritis is another one, and then neuritis of an unknown etiology. So just quick, and there's enteric neuropathies that are iatrogenic or induced by the treatment that we provide to patients, including drug treatment. These include anti-neoplastic treatments such as vincyclic alkaloid and cisplatin. Postoperative ileus is a well-established phenomenon, very poorly understood, the mechanism of that. And then intestinal transplantation can induce a neuropathy or of ischemia. And then opiate-induced constipation is usually caused as we treat patients with chronic opioids. The constipation is a constant problem that you have to deal with clinically. So quickly, I'm just going to show some of these diagnostic images of some of these conditions. This is called diffuse esophageal spasm. This classic corkscrew esophagus resulting from the spastic contraction of the circular muscle in the esophageal wall. And although it's called the circular muscle, it's actually a tightly spiral helical configuration around the esophagus. And the longitudinal muscle is not really straight up and down. It's a much more longer helical spiral. So they're both really helical spirals, but one's tighter and one's much more loose. And they're called circular and longitudinal. And because of that, the way it contracts produces this corkscrew type arrangement in this diffuse esophageal spasm. And these are the newer way of looking at esophageal function with these heat maps. And I think if you're a gastroenterologist, you understand these. I still have a hard time. These came up a couple of years ago, and I still haven't been able to get my head around how this is done. This is time on the x-axis, and then on the y-axis is pressure, and then the colors refer to. I'm sorry. The colors refer to pressure, so, and then the y-axis is distance along the esophagus. So you're seeing what happens during, for example, swallow. What happens to the pressure along the esophagus from the pharynx to the lower esophageal high pressure zone, or the sphincter. And this was happens with achalasia, with compression. You have these contractions of the entire esophagus. And here's spastic achalasia. The color isn't indicative of the pressure in these maps. I still have a hard time getting my head around these heat maps of pressure waves in the gut. So Let me move on to the bladder and urethra now for the rest of the talk, because that's really where I spent most of the last 30 years of my life, on bladder and urethra. In the United States, there's an estimated 34 million men and women with overactive bladder, and the costs are estimated at $19.5 billion for and so this is $19.5 billion for incontinence and $12.6 billion for the overactive bladder, and that seems like a lot of money. But if you calculate how much that is compared to the total U.S. healthcare annual spending, it's only 0.8%, but still it's a lot of money, and it's a lot of people that are affected by these conditions of overactive bladder and incontinence. And the patients that you treat with spinal cord injury, the bladder issues are a major issue to their quality of life. So I want to quickly go through some of the what's known about bladder function. When the infant is born, the bladder is sort of automatic. The spinal cord control of bladder is the major way that the infant empties the bladder. And if some of you talked about, if you have young kids, if you're changing a diaper, often when you stimulate the perineum or even the genitals, you'll induce a reflex micturition or even defecation, and that's because of this reflex that's overactive in infants. And then with maturation, you develop a suppression of those by upper centers from the pontine micturition center down through to the sacral centers that control this reflex. With conditions that cause incontinence, there seems to be sort of a reversion back to this involuntary neonatal reflex, and our goal in therapy is try to take that and move it back towards voluntary control. And we're making some progress on this. And this came from Campbell's Urology from Dr. Mike Chancellor and Naoki Yoshimura at Pittsburgh chapter. And I'm glad to see they finally read some of our papers in this. I'm going to take you through some of the findings that we have on how the actual smooth muscle of the bladder contracts. It goes from this distended state to a contracted state, and the major mechanism is through muscarinic receptors, acetylcholine released from the parasympathetic pelvic nerve, activating GTP binding proteins and phospholipase C and alkyl triphosphate, acting on the sarcoplasmic reticulum to release calcium, that activates the calmodulin and activates the actinomyosin contractile proteins. Now, this is a very simplistic way of looking at it. It's much more complicated than this. And the bladder, in the last 10 years, there's been a intense investigation into the role of the urothelium and its sensory role in bladder function. It releases a number of substances, in particular ATP and acetylcholine, that act on the underlying suburothelial, not smooth muscle cells, but these interstitial cells of Cajal-like cells that are in the bladder, that are in electrical communication with the smooth muscle cells. So the new paradigm, the new thinking of how signal transduction occurs in the bladder is that these c-KIT positive interstitial cells are the ones that are the pacemaker, or also known as the pacemaker cells. They don't contract themselves, but they respond to the released neurotransmitters and neuromodulators to mediate the smooth muscle contraction. And that's an important difference in our thinking now of how to control smooth muscle contraction that's overactive, because it's not just the smooth muscle that you need to think about, it's these interstitial cells and the substances that are released from the urothelium. So the idea is that there's a number of things that are released from the urothelium, including these substances such as bradykinin, the neurotropins, tract A and P75, receptor activating substances like neurokinins, norepinephrine, the activating alpha and beta adrenergic receptors, and acetylcholine that can activate both nicotinic and muscarinic receptors on these smooth muscle and the interstitial cells of Col-like cells. These protease activating receptors and a number of these channels, such as the sodium channels, these amiloride sensitive sodium channels, and the TRP, the transient receptor potential channels. There's a whole class of them that are now known to be involved in the in this mechanism in the bladder. So this is a little more complicated role of the signal transduction that occurs in, and it's not, and based on our data that we produced in the last 30 years, it's not just M3 receptors. And finally, I think it's made it to the Journal of Urology with Mike Chancellor and Naoki Noshimura actually read our papers and described that the M2 receptor does also play a role. But as far as clinical practice goes, it's probably irrelevant with the M2 or the M3 receptor, what it does, because the antagonists that are used to treat overactive bladder, the muscarinic receptor antagonists, act on the M2 or the M3 receptor doesn't seem to make any difference in their clinical efficacy. And I've always thought that's probably because that the target is probably not the bladder itself. It's probably the target for antimuscarinic therapy for overactive bladder is probably other parts of the body that are involved in control of the micturition response, including and these receptors are everywhere in the spinal cord, and the onus nucleus controls the reflex in the pontine micturition center, and even in the higher cortical centers that are involved in control of bladder function. And it seems to me that these receptors in these other areas are as important, if not more important, than the ones that are actually in the urinary bladder itself, and maybe the target of this therapy. Let me just go on to talk a little bit about the actual sensory innervation of the bladder. There's two, and the genital system. There's two major nerves. The hypogastric nerve is primary sympathetic, coming from the inferior mesenteric ganglion that receives sympathetic fibers from both the bladder as well as the genitals and the prostate and the sphincters. And they go through this major pelvic plexus that lies right next to the entry of the ureters into the bladder. And then there's the sensory nerves that follow the motor fibers through the pelvic nerve as well as the pudendal nerve. And these are going through a different spinal cord segment there in the primarily S2 and S3 in the human, whereas the sympathetics come all up and down the lumbar cord. This is just a cross section of the sacral spinal cord, showing the location of these different afferent components that represent the bladder, urethra, and the genitals, where the component, the cutaneous perineal afferent components represent the afferent fibers innervating the perineal skin contained in the pudendal nerve. And then there's also muscle spindle afferents components that are represent these large 1A and B afferent fibers that are contained in the actual skeletal muscle components, including the levator ani and the actual muscle spindles in the sphincter muscles. So I want to spend a little bit of time talking about development of drugs, and these are a list of drugs that are used that have actions on the bladder and have been used to treat these overactive bladder conditions, including anticholinergics. The latest one is tolterodine. In vitro, it has exactly the same M2 and M3 selectivity as atropine, but it seemed to have much more selectivity for the bladder over the salivary glands in vivo. And it still has not been adequately explained why that is. But certainly patients that take toldaridine don't have complain as much dry mouth as they do if they're taking the classic anti-muscarinic drug, atropine. And then there's smooth muscle relaxants that I'm sure you're very familiar with, like soclamine, fluvoxate. There's calcium channel antagonists that are classically used to treat high blood pressure, that can be used to treat overactive bladder. And then the potassium channel openers, chromokalin and panacetyl. And these prostaglandin synthesis inhibitors have been tried in a number of studies, and they have been various degrees of success. And pretty much all these drugs have limited penetration in terms of the ability to successfully treat all patients. So I think that may be reflective of the fact that we don't understand the mechanism of disease, and the mechanism of disease in these patients is probably different from patient to patient. So drug therapy probably needs to be tailored to the patient, and so it's sort of a trial and error that physicians have to go through to try to understand which one works best for which patient. And I think some of this may be that as a result of this, I think sort of a fallacy in thinking that an underlying assumption over the past has been that drugs that affect the normal physiologic mechanism will likewise affect the pathophysiologic mechanism. So we know that acetylcholine causes the bladder to contract. So, and that because muscarinic receptors are activated. So if we inhibit muscarinic receptors, we should be able to inhibit the overactive bladder. But we really don't have a mechanism for the pathophysiology of a lot of these conditions, including overactive bladder. So does it really make sense that the pathophysiologic mechanism will be the same as the normal physiologic mechanism? I propose that if that's true, if they're the same, then what makes it pathologic in the first place? You know? So I think the anti-muscarinic treatment of overactive bladder is based on that fallacy that inhibiting the normal mechanism is going to inhibit the pathophysiologic mechanism. And I think it doesn't. I think it acts different ways, probably through the central nervous system and spinal mechanism involved in bladder function. I want to talk a little bit about another drug that has been used more recently and to a much more greater extent, botulinum toxin. This is a schematic diagram of the bladder. Here's the bladder here, and we're looking real close at the synaptic connection between the nerve fiber and the postsynaptic membrane, where there's these snare-type proteins, the synaptobrevin in the vesicle that contains acetylcholine, and here's the SNAP and syntaxin membrane proteins in the presynaptic membrane. So when the cell is depolarized and these vesicles fuse with the presynaptic membrane, it's through this connection between the connection between these synaptobrevin and these SNAP proteins. And then what that does is allow fusion of the vesicle with the presynaptic membrane and release of the contents and activating of the postsynaptic receptor mechanisms. But it's these snare proteins that the botulinum toxins act on. They are taken up by the presynaptic membrane, and then they act on the these snare proteins and inhibit this mechanism. Now, there's lots of different isoforms of botulinum toxin, and they each have different effects, but they all result in the inhibition of this fusion of the presynaptic membrane of the synaptic vessels containing acetylcholine with the presynaptic membrane. So, and that's been getting a lot of use now in patients with overactive bladder and sphincter function that needs to be suppressed. So let me talk a little bit about the mechanism underlying plasticity and bladder reflex that are induced by different pathologies, because I want to spend the last half hour, if I can, on the findings that we've had with reinnervation of the bladder. So in different conditions in rats, primarily, including spinal cord injury, urethral outlet obstruction that causes hypertrophy of the bladder, chronic inflammation models, and even bladder denervation, they exhibit this increased level of neurotrophic factors, neurokinins, that are released from this bladder smooth muscle wall. And these increase the excitability of the C fibers, the pain receptors, and alter the reflex mechanisms and the parasympathetic ganglion, and as well as the effects on the sympathetic pathways through the hypogastric nerve up here. And they're organized in the spinal cord in two different places, one in the sacral cord, and another up in the lumbar cord where the sympathetics come in. So there's been a lot of work that's been done primarily in rats, and a lot of this is done in Pittsburgh with they have a huge project on trying to understand the effects of outlet obstruction and spinal cord injury on bladder function, and primarily as a rat. Excitatory alpha-1 adrenoceptors, one of the major findings is that's upregulated. That the upregulation of these alpha-1 mechanisms provides additional excitatory input to the bladder, maybe somewhat explaining this overactive bladder from these different models. So I want to take a look, like a 5-minute break, and then maybe we can talk about the findings that we have with bladder, and maybe take some questions during that time. So this, how do I get to the, I want to get to that. I don't know how to use this Mac very well. I have another.
So you're done with this PowerPoint?
Yeah, I have another one on that same drive. It's called number 2. Yeah, day 2. How'd you do that? How the hell did you do that? Yeah, that's the one. I couldn't get to it.
No, it was just hidden
I'm
behind
not
the
real
other.
good at these touch screens. I'm
Sure.
an old school mouse guy, you know. Okay, there we go. Okay, so I wanted to see if I could tell you a little bit about what we've been doing with dogs. And I think you, as I'm going to try to get started again, guys. You get a chance. There is another person going to give a talk right at 10. Is that right? Yeah. So I wanted to see if I can get through this. And I think this, for your clinical practice and taking care of spinal cord injured patients, I think what I'm going to say in the next few minutes is probably going to be most relevant to your clinical practice, particularly in the future. We've been trying to understand how to treat the lower motor neuron lesion bladder for the last 10 years in collaboration initially with a group from Shriners Hospital, really headed by Randy Betts at the time. He's semi-retired now. But we had a whole series of collaborators over the years on this project. And it really takes an army to do this, because this is a major undertaking to try to develop surgical approaches, neurosurgical approaches to treat the lower motor neuron lesion bladder. And really, the thing that got me interested in this is that a paper by a scientist named Kim Anderson. Now, she's an interesting case where at age 18, she had an auto accident that left her paraplegic. And she still finished college and finished graduate school, and actually is working out at the UCLA Center for Spinal Injury. And she's done a lot of work with nerve growth factors, and a lot of other things over the years. But the most interesting thing that she's published in my mind, from my point of view, is this paper in Journal of Neurotrauma, where she surveyed patients that have spinal injuries and just asked them, what's most important to you? What do you need? What do you need us to restore? And if they were quadriplegic, regaining hand and arm function was most important. That's no surprise. Of course, they can't move their hands. That's important to them. But if they're paraplegic, regaining, the most important one was regaining sexual function, not walking, because they can get around in a wheelchair. And improving bladder and bowel function was of shared importance to both, and both are greater than walking. So all this work that we've been doing to try to get these spinal cord injury patients to walk again, that's not the most important thing to them. And there really hasn't been a lot of work on how to restore sexual bladder and bowel function in these patients. So that's what's got me interested in this. Now here's a diagram of the innervation of the bladder showing the two different major pathways. Here's the pudendal nerve coming in to innervate the three structures. The pudendal nerve innervates. It innervates the external urethral striated muscle sphincter, the anal sphincter, and the contractile tissues of both the corpus cavernosus in the human and the erectile tissue in the clitoris of the female. And then there's the pelvic nerve. Both of these go through the major pelvic plexus that innervates the smooth muscle structures in the bladder, as well as some of the urethra also. And then there's these hypogastric nerves that come up here through what we talked about earlier. So I think you as PM&R physicians are well versed in the difference between upper and lower motor neuron lesions. I'm not going to spend too much time on it, but as far as the bladder is concerned, the lower motor neuron lesion that's below this T12 to L1 vertebral level, it affects these cord segments that are involved in bladder function. and includes actually conus medullaris lesions themselves, as well as lesions to the cauda equina. Often happens in a car accident with a lap belt injury where the entire conus and the cauda equina is avulsed, pulled out from the spinal cord. And so it results in a flaccid bladder paralysis. And we did a little survey over a number of years at Shriners Hospital. At least 25% of the patients at Shriners have this kind of lower motor neuron lesion bladder. There are ways of treating the upper motor neuron lesions. There's nerve stimulation techniques that are various degrees of success, but we wanted to tackle this lower motor neuron lesion because these patients really have no connections between the central nervous system and their bladder. And so we need to establish new connections in order to restore function. So one of the first things we did was we cut the nerve that caused the bladder to contract in these dogs and sewed it back together and placed a tripolar electrode proximal to that nerve anastomosis site. And then we actually put a nerve cuff or a cuff around that anastomosis site and pumped an osmotic pump, pumped in nerve growth factors to see if we could stimulate additional growth. That turned out to be a bad idea because these growth factors are secreted by the denervated organ, and there's a concentration gradient that's established that the new neural pathway follows the concentration gradient. And supplying it, turns out that we found out that supplying it like this only led to an increased neuroma formation. The nerve growth factor worked very well, but the nerves grew back up towards the source of the nerve growth factor and not what we wanted it to go towards the bladder. So I think nerve growth factors are important, but we don't know how to apply them. And so the third aim would be to bypass the nerve transaction, achieving emptying by electrical stimulation of with the new nerve pathway. And so this shows functional electrical stimulation after where we just did a transection and a root repair. And so 78 days post-operative stimulated nerve had no effect, whereas up to a year later, we did get an effect on the right side, but on the left side, we didn't. So, and then we did these neurotracing studies where you inject these vital dyes that are taken up by the nerve in vivo and transported back to where the cell body exists. We inject it into the bladder and look for it in the spinal cord. And this is done with Dr. Mary Barb, who's the neuroanatomist. What we also did is we took the nerve surgery site where we did the actual transfer, and then you can use these lipophilic dyes post-mortem in fixed tissue, where you can place it on either side and trace them across the nerve anastomosis site to see if these nerves are actually grown back together. And this shows some of those findings of the reinnervation of the bladder, showing these vital dyes being taken up and deposited into the spinal cord of these dogs in cell bodies that are different from the natural cell bodies in the sacral cord. And here's the showing that this actually proves that at the anastomosis site, these nerves are actually growing across the site. Whereas you put a red dye on one side and the green dye on the other, they actually co-localize, and proving that the nerves have grown across the site. So we did this. So we decided to use a nerve that runs right along the pelvic plexus to reinnervate the bladder. So we did these the spinal transection here, where we actually transected the sacral nerves that cause bladder. So we stimulate the nerves until we get a bladder contraction. Those are the ones we cut. And in a dog, it's usually S2 and S3 on the S1 usually, S1, 2, and 3. And more recently, we've been cutting everything below from S1 all the way down to get a complete decentralization of the bladder. And then we use this genitofemoral nerve that is really only involved in like thermoregulation of the testicle. It's the cremasteric reflex. It's not that important a nerve. Patients would much rather have their bladder function than thermoregulate their testicle. So we transferred this to calling the anterior vesical branch of the pelvic nerve. Now, I finally, it took me a long time to realize that when urologists think of the pelvic nerve, they're thinking of the nerve that comes from the spinal cord into the pelvic plexus. That's what they're calling the pelvic nerve. That nerve, when it gets to the pelvic plexus, it branches and it goes to a number of different sites, not just the bladder, including the urethra and the contractile and the erectile tissue of the penis. And so we did have a patient that was a lot likely would benefit from this surgery, but our collaborating urologist did not want to do the surgery because didn't want to interfere with potential erections. Having done his postdoc in Boston, his fellowship and stimulating what he called the pelvic nerve in dogs and producing erections. But it's the pelvic nerve comes from the spinal cord to the pelvic plexus that he was doing up there with Mike Soroka in Boston. What we're doing is we're transferring to the branch of the pelvic nerve that goes directly to the bladder. So it can't go to the contractile tissues of the urethra or the erectile tissue. And so we're calling it a different name now so that we distinguish it. So urologists aren't afraid to do the surgery, but they're still afraid to do the surgery. So I think neurosurgeons are much more likely to be interested in doing the surgery than urologists because they don't want to mess around with the pelvic plexus. Just, bad things can happen down there. But neurosurgeons, on the other hand, are much more likely to be involved in this. And I think maybe a place like Moss Rehab will be a place where this kind of surgery might take place in humans for the first time, because I think we're pretty close to being ready for that. Let me just take you through quickly. I only have 10 minutes left, so I'm not going to be able to spend a whole lot of time on all these slides. But this was the timeline of the design of the first study. Is that right? I can take the little extra time? Okay. So what we did, and this is the 3 years of one of the initial studies, and this is how you have to design these studies because we have a limited number of animals we can house at one time, so we have to kind of stage it so that the surgeries and the euthanasia procedures and the injection of the fluoral gold that dies to try to show retrograde tracing are done in a way that we can get everything done in 3 years. So this is how we designed it. Now, one of the first studies we did, Randy Bett's idea was to use an intercostal nerve and move that along the spine into the sacral plexus to anastomose to the S2 route that goes to the bladder. And we've shown in the first dog that wasn't possible. We actually had to use 2 intercostal nerves to be able to reach, and we had to paste them together. It actually worked on that side, so but we almost lost that dog on the table. So we quit while we were ahead, 100% success rate on that side. But we decided that during that operation, instead of using on the other side, instead of using the intercostal nerve, we used a nerve that innervates the tail, and that's also a somatic nerve. So we transferred that to the other side, S2 route that goes to the bladder. And then we did that in a number of, so that's called CG coccygeal nerve transfer, coccygeal nerve. And then we did a number of dogs where we did the denervation or decentralization, and then immediately transferred the genitofemoral nerve to the anterior vesical branch of the pelvic nerve. And then in a number of dogs, we separated the decentralization from the reinnervation, because if you have a patient that comes in with an accident, you're not going to do this transfer surgery right away. You're going to probably wait to see if they get better on their own. And we talked to the physicians that are treating these patients, Randy Betts in particular, and he says, if after 3 months the patient doesn't get better, they're probably not going to get better. Their bladder function is not going to return. So we did 1 and 3 months delay between the decentralization and the reinnervation. And then we had these electrodes planted, and we stimulated them to look for return of function. And then we injected the dyes and looked for reinnervation. And at the euthanasia procedure, this is the intraoperative view. Let me just give you a little perspective here. The tail of the dog is down this way. The head is up this way. This is the Foley catheter that's inside the bladder that we did a vesicostomy to drain the bladder during the postoperative period, and that's how we catheterize, and we're measuring bladder pressure through this Foley catheter. And here's a Medtronic's probe electrode unipolar. The other ground is to the Bovie ground, stimulating this anterior vesical branch of the pelvic nerve that goes to the bladder. The pelvic nerve that urologists talk about is buried down here, coming from the spinal cord into this pelvic plexus. We're not doing a surgery to that nerve, which urologists do not want to mess with, because bad things can happen. But we've proven in a number of dogs now that this branch of the anterior vesical branch is certainly amenable to this surgery. And these are the general results of our first round of series. We got return of function by stimulating the transferred nerve in 3 of the 3 animals with the coccygeal nerve transfer, 3 of the 4 animals with the immediate genital femoral nerve transfer, and 3 out of 4 animals with a 1-month delay between the decentralization and the reinnervation, and actually 5 out of 6 animals with 3 months. So these are very encouraging results that we had years ago, and we published this in 2008. But it really hasn't caught on. Surgeons haven't decided to do this. So we continue to do these studies with the retrograde tracing, and Mary Barber actually counted the number of positive cells per cross-section of the spinal cord at the lumbar, sacral, and coccygeal level. And so sham is what's normal. So if you inject the bladder with fluoro gold, 3 weeks later, look at the sacral cord, and almost all the fibers from the bladder are in the sacral cord. We couldn't measure any in the lumbar or coccygeal cord. With the root repair, the same thing is true. But with the genital femoral nerve transfer, they were all primarily in the lumbar cord. These few that are still in the sacral cord is probably because we didn't sever all of those sacral roots initially. And then, of course, the coccygeal nerve transfer, a number of fibers appeared in the coccygeal spinal segments. So then we asked with the question of when you're transferring these nerves, this new pathway that's developed, does it innervate the bladder muscle directly, or does it innervate these ganglion cells in the bladder wall? So are nerves or muscle reinnervated by a new pathway? And so the way this is done is these lipophilic dyes. Post-mortem, you put the dye directly on the transferred nerve between the spinal cord and where the transfer was done. So the dye has to go through the actual anastomosis to get to the bladder. And this is done post-mortem because it's a lipophilic dye that diffuses across the lipid bilayer of the nerve, and with enough time incubation in vitro and post-mortem, it'll get all the way to the actual where it ends in the bladder wall. And these are some of those pictures showing that in the normal dog, putting dye onto that nerve, it only innervates these the muscle cells. These are the muscle fibers here, and here's showing the dye on there. The PGP 9.5 is a stain that pan sham with PG 9.5, but in the root repair as well as the genital femoral nerve transfer and the coccygeal nerve transfer, the new nerves innervate both the muscle directly as well as the intramural ganglion cells. So this just shows a quantitative result of that study, showing that basically the bottom line is that reinnervation from a somatic nerve that formerly innervated the skeletal muscle, such as the coccygeal nerve, preferentially results in a direct reinnervation of the bladder muscle cells over the indirect bladder reinnervation via the intramural ganglion cells. And as to whether those cells are functional, those muscle cells that are directly innervated by a new pathway versus the intramural ganglion cell being reinnervated, which are more functional, we don't really have a good answer to that. So we're really encouraged by these findings, showing that reinnervation can be achieved by immediate repair or coccygeal nerve transfer or genitofemoral nerve transfer at the time of denervation, or genitofemoral nerve transfer either 1 month or 3 months after bladder decentralization. So these are very encouraging results. So the reinnervation with the same nerve, just cut the nerve and sew it back together, or either a primary somatic nerve, such as the coccygeal nerve, or primarily sensory nerve, such as the genitofemoral nerve. These new pathways, when they grow in, they don't know which nerve, which to innervate, so they innervate both the ganglia as well as the indirect, directly innervate the detrusal muscle. Now, we've also have to deal with the outlet. And the pudendal nerve is the one that deals with that innervates the skeletal muscle of the outlet. And it subserves the 3 major functions that patients want to regain. That's sexual function, at least erectile function in male, and erectile function in females. And urethral and anal sphincter continence. So we did a cadaver study to see whether this anterior femoral nerve that runs down the inner thigh will be able to reach to the ischial tuberosity where the pudendal nerve emerges from this pudendal canal, also called Alcock's canal. And we showed and published this in the Journal of Neurosurgery that this is certainly feasible in the human. And I'm not going to go through all these slides because we don't have time for this. We did a really detailed anatomic dissection in the anatomy lab, showing that these branches are able to be harvested and they'll reach to the pudendal canal and can be used to actually reinnervate the pudendal nerve as it emerges from Alcock's canal there. And this is showing there's a diagram of this femoral nerve that we actually moved. Here it is. In the actual photograph of the cadaver, and here's the femoral nerve moving down to Alcock's canal. It's able to reach there. Then we did the same thing in a cadaver dog, and the cadaver dog was much more difficult than human. Pretty much all the surgery we do in dogs is going to be much more difficult than human. And we were able to get this nerve and measure it, and it was able to reach down to the pudendal canal in the dog. And then we actually did this surgery in 3 dogs, where we cut the nerves that innervate the bladder and the sacral cord, and then did the femoral nerve to pudendal nerve transfer. And we probably okay. Let me just. At that point, we also had a partnership with the Alfred Mann Foundation that produced these bions that are radio frequency activated stimulators. So these little devices that are basically, they can miniaturize it now, I think, to the size of a large rice grain. So it's a very small device, and the ones that we had were actually were leaded, so we could put a nerve cuff around it. And so one pole is the actual on the bion, the other pole of the electrode is around the nerve cuff. So we put that around the transferred nerve, and everything's implanted. And so this is an external coil that's placed over the bion that's put near the skin, and then you can activate it to cause this kind of stimulation. And the current is balanced because you don't want to have current going in one direction, or you'll get your electrode will start to electroplate onto the tissue. So it's balanced. The area under this curve is the same as the area under this curve, but you actually get stimulation by this pulse. And here's what happened in our dog number 2, 208 days after surgery, where we activated the bion on the left side, and we got an increase in urethral sphincter pressure and anal sphincter pressure on this bion with a nerve cuff around the transferred femoral nerve that was transferred to the pudendal nerve in Alcock's canal. And here is just a curve showing increasing stimulation intensity with different times postoperatively, showing a sort of a recruitment curve of the more intense the stimulation, the more number of muscle fibers are activated in both the urethral and the anal sphincter. So in these 3 dogs, we got very positive results in this transfer procedure. So we have a way of reactivating both the bladder with genitofemoral nerve transfer and the urethral and anal sphincter with femoral to pudendal nerve transfer. I don't think I'm going to spend too much time on this. We don't have too much time, but suffice to say we did the retrograde tracing on these studies, and we also showed that these structures were reinnervated by the transferred nerves, and the evidence is that it's not only a different part of the spinal cord the retrograde dyes go back to, but it's a different lamina in the spinal cord itself. And she actually did count this as showing that in the normal controls, most of the, in this case, we injected the anal sphincter with a different colored dye and the urethral sphincter with another colored dye and the bladder. So we had 3 different colored dyes. And you can see that in the normal controls, S1, 2, and 3 are the only structures that have fibers that go to the bladder, urethral sphincter, and anal sphincter. But when we did the femoral nerve transfer, it's the L2 or L1, which is with the origin of the femoral nerve. Let me just skip over this and show what happens when we reverse the vesicosity. So the bladder function in these patients, generally urologists like to create a vesicotomy so that the bladder can empty, especially if the urethra is competent and closed. And so we did that in these dogs, and one of the dogs that we did the genitofemoral nerve transfer, we found that this result with the bion activation, we actually did get increased detrusor pressure 26 weeks after the surgery. And so we decided to reverse the vesicotomy in this dog. So the question is this functional reinnervation or not? What we're trying to answer. So we in dogs, it's if anybody has pets dogs, they assume a specific posture when they do micturition, especially if it's a female dog, they do this behavior. Male dog lifts their leg, but we pretty much all use female dogs in this project because they're easier to catheterize. And this posture when they defecate. So we had and I actually put a picture of this in my grant, this dog squat procedure. So I'm really surprised that they actually funded it, because I showed a picture of a dog doing this in the grant, but I'm not going to complain. And we had a camera over their cages, and we actually measured the time that they spent doing these different postures. And it turns out that genitofemoral nerve transfer dogs had more voiding events per day, but the duration was the same. Whereas the femoral nerve transfer to pudendal nerve, they had increased number of time for defecation. So I'm not exactly sure what this means, but this may be related to functional denervation. But the dog that we reinnervated, I don't know if this is going to play here. Well, okay, it's not going to play here. She got out after the procedure and walked across the lab floor and emptied their bladder. And so we've actually reinnervated the dog. And so,
It might not work.
is that it? Yeah, I may
No,
not have
it's
the, I may not have the movie file on this stick. This is the problem going from the PC to the Apple. So I just want to go through quickly. These are a number of photographs of our cadaver study of the genitofemoral nerve transfer and the femoral nerve transfer. Let me just go to one of the things that Justin Brown, the neurosurgeon that's been working with us on this thought might be a good idea is to, when you take the femoral nerve and transfer it to either the bladder or the pudendal nerve, run it close to the skin so that if it doesn't take over function on its own, you might be able to use transepidermal nerve stimulation to activate that nerve. We've tried that in dogs. Dog skin is not the same as human skin, and it was much harder to get the electrical stimulation of the femoral nerve that ran close to the skin of the dog. We did get in a few dogs, but I think in humans it might be much easier. And so we compared the femoral nerve branch to the genitofemoral nerve, which is primary sensory. And let me just go through that and tell you quickly that a donor nerve that has primarily somatic nerves in it has much better reinnervation potential than the genitofemoral nerve is primarily a sensory nerve. And that was sort of a hypothesis that everybody thought was true. We actually proved it. It took us a long time to prove it. But then we also looked at retrograde dyes injected into the clitoris in these dogs to see if what happened, because we were reading, I read some papers where patients with complete Asia A spinal cord injury had evidence that and this was done in New Jersey with an fMRI study, where they showed that cervical self-stimulation is associated with activation of the brain areas involved in the vagus nerve. So the idea is that these patients are actually feeling sexual stimulation through the vagus nerve. So we thought, well, maybe that's happening in our dog. And actually, they even reported, actually sexual orgasm in the scanner during self-stimulation, and documented with fMRI activation of these vagal areas of the brain. So we did this in our dogs, and lo and behold, if you look at the unoperated controls, you see absolutely no retrograde tracing from the clitoris or the bladder and this was in the nodose ganglion, the sensory nucleus of the vagus. But in the genitofemoral or femoral nerve transfer, in both the nodose ganglion as well as the motor, the dorsal motor nucleus of the vagus, a number of fibers were activated. So what we think this means, and I'm going to have to stop soon because I'm going to get the yank here, is that the vagus nerve, it's actually sprouting and reinnervating the structure that it normally doesn't innervate. And so I think that may be true for other, but it didn't seem to innervate the urethral sphincter or the anal sphincter, but it did reinnervate the bladder and the clitoris. But we didn't see a general increase in sprouting. The sympathetics in the sympathetic chain ganglion, we measured them also. And there were no statistically significant increase. There was generally an increase over in these different nerve transfer, but these weren't statistically significant. So it's not just a general increase in sprouting, it's specifically for the parasympathetic fibers of the vagus nerve that sprouts and reinnervates other structures. So this is the normal innervation of the vagus to the viscera, and then with decentralization that we created with the pelvic nerve, we get this sprouting to reinnervate the bladder and clitoris. I think I'm going to stop there and let the next speaker, so I don't want to dominate too much of your time. But I really think that this next set of experiments that we're going to be doing in dogs is to reinnervate both the pudendal nerve as well as the bladder. So we're using the femoral nerve transferred to the Alcock's canal, as well as, in this case, we're going to use primarily a somatic nerve, the obturator nerve, that runs in the same pathway as the ilioinguinal, as well as the genitofemoral, and transfer that to the anterior vesical branch of the pelvic nerve in the same animal, and see if we can get true control of bladder function, both storing and emptying function. And we've done surgeries on 11 dogs now. In the first surgery for the transfer of, we do the pudendal to the femoral to pudendal nerve transfer first, and then 3 weeks later, because we can't do it in the same operation, it just takes too long. The first time we're going to do the obturator to vesical branch of the pelvic nerve transfer will be next Wednesday, actually. So now we'll have dogs that have what we've proposed for the human type of surgery for this. And so depending on the results, I think in a year or two, we'll be ready to think about how to do this in a clinical trial in patients, a clinical surgical trial. And I think a place here like Moss Rehab has a high potential of being a place where this kind of cutting edge surgical reinnervation can start. And I'd be interested in anybody here that's interested in this kind of frontier translational research. Okay, I think that's all I have to say. Thank you.